Hi, welcome to Unit 4, Chapter 3, where we will be reviewing phylogeny. As always, if you click on this book, you will get a copy of the guided notes. Now, when we're talking about phylogeny, we're talking about the history of life on Earth in relationship to evolution. So last week we talked about evolution as one generation changes into the next generation. But now we're going to follow those changes over thousands, if not millions of years. Now, if I take all of the Earth's history and I condense it into one 24-hour period. So we have 24 hours. So it starts on midnight, and then we're going to go all the way around to midnight again. So we're going to start at midnight and end at midnight. When do you think that Homo sapiens kind of showed up? When do we see the first evidence of modern humans? That means modern humans with our modern sized brains. And here we are right here. Now, if I were to condense all of Earth's history into one 24-hour period, humans have only been around since about 1159. So we've only been around for a very, very small amount of time compared to all of these other organisms. So let's look at some of those other organisms and some patterns that we see, broad patterns that we see throughout history. One of the ways that we have geological history, or one of the ways that we have this history of evolution, is through the fossil record. Now, the fossil record is notoriously incomplete. That is because fossils develop over a long period of time under very specific conditions. The animal had to have died intact, been buried in sediment, and then had some water flow, which will remove the organic material and replace it very, very slowly with minerals. So when you find a fossil, it is actually rock. It's made of rock. It's not made of organic material anymore. So let's look at some of those examples. Now, when we are looking for fossils, we're digging. And the top layer of dirt is always going to be the newest layer of dirt. So as you dig further and further down, you are getting older and older fossils. So it creates this nice, beautiful image. But that's not always what happens in real life. The earth moves, okay? It's dynamic, and we have rock slides, we have mountains, we have, other types of borders. And sometimes the rock kind of slides over and it's tilted. And in that case, you would have the oldest rock at, this layer, the bottom layer. And then as you move to the side, you're kind of walking through history into the future. And the newest rock would be on the top here, usually exposed. So there are different shapes, but generally the oldest fossils will be found at the very, very bottom, and the newest fossils will be found at the top. Now, if you want to click on this little game here, down at the bottom it says layers of time. We can start this game, put it on easy. I don't want to put a lot of effort into this game, but what we're going to do is we're going to find out the geological time that these organisms were alive. It already gives us a clue about the oldest layer, so I'm going to move that to the bottom. Now, we would expect some changes throughout history, but not too many changes. So we're going to organize these organisms based on appearance because we're not able to extract any genetic information from these organisms. They're not organic. They are minerals. Now, I have all of these grouped together, but I notice a little gap here. So I don't think that these go right next to each other. And I also see these feathers here and these dinosaurs right here. So we have birds and dinosaurs. These layers do not have one of these little seashells. So they are probably at the top because I know that this is the oldest layer. Since this also has a dinosaur and a feather, it's probably going to be the top layer here. And then I know just based on my prior knowledge that dinosaurs were not living in modern day, but birds are. So I'm going to assume that this is probably the top layer. I have a feather here as well, which might go here. And then I have these guys that are grouped together, and I can check my work, and it looks like everything is correct. So my oldest layer are going to be my oldest fossils, and my top layer is going to be my modern animals. Let's move forward. If the rocks and the fossils are so unreliable and we can't date based on the sedimentary rock that's found around the organism, then we can do something called radiometric dating. Radiometric dating relies on the decay of isotopes. Isotopes are different versions of an element, for instance, carbon, that's maybe a little bit heavier. It has an extra neutron. And so it doesn't change the identity, but it does change the molecular properties in the way that carbon 14 is not as stable as some other versions or other isotopes of carbon. Carbon is found in all organic material, and so it's a really easy one to measure when we find an organism that still has organic material in it. Now, while an organism is living, I would expect to find 100% of their organic carbon left. Once an animal dies, it starts to decay. If I measure an organism and it has about 50% carbon left, that means I have met the half-life. The carbon will be decaying the entire time, but by the time about 50% of the carbon is gone, it's been 5,730 years. If I measure an organism and it has 25% of the carbon 14 left, then I can assume it has been two half-lives and it's been about 12,000 years. Now, if I get all the way down to this end of the spectrum here, carbon dating does not continue to be useful because here I would have 0% of the carbon 14 left and I would have to switch to a different metric. Either none of the organic material is left or it's so old that it's degraded and we can't measure it. So we can switch to a different isotope. Another isotope that is sometimes used is uranium. Uranium decays extremely slowly, though. It can be useful because it's found in non-organic materials, but it is so slow that you would have to be measuring something that's like 9 billion years old to be able to find any important data because it takes 4.5 billion years for uranium 238 to degrade into lead. So if you want to click on this image, we are going to go to the PhET simulation. I'm sorry, it does take a minute for it to load, but this is a really great simulation because it emulates carbon dating. The first page, the landing page here, it's just talking about these different isotopes. So if I have this carbon 14 isotope and I just fill the page with it, notice that it degrades into nitrogen kind of randomly. But about 50% of it has degraded by the time it reaches its half-life of 5,730 years. The same thing with uranium. If I add all the uranium in here, it will slowly start to decay into lead. And by the time about 50% of it has decayed, we have reached the half-life of 4.5 billion years, which is just impractical. So mostly we're going to be using a custom decay where we get to determine what the half-life is. So next I want you to go to the dating game. This is not speed dating, this is carbon dating. I am currently selecting carbon 14 isotope, and so I'm going to be measuring how much carbon 14 is left in these samples. So I would expect the newest organisms to be towards the top and the oldest organisms to be towards the bottom. By the time I get to this third layer here, there's no carbon left. So I'm going to have to switch to a different metric, but let's do the top layers first. Now, remember, if an organism is still alive, it should have 100% of its carbon 14. If it's dead, it will slowly start to degrade. In order to play this game, we will have to cross-reference the amount of carbon that's left with our carbon data right here. So here we have it set for percent, and we have 98.2% of the carbon left. So I'm going to move this until we are at 98.2, which is just a whole lot. It's almost fresh. 98.2 says 151 years, and it accepts my answer. All right, let's measure something that's more dead, like this wooden cup. Since it's made of wood, I know that there's some organic material in it, and it measures at 88.2. So you're just going to drag this over until you get close to 88.2. It's not perfect, but it'll accept that 1,026 years. As I move further down, I'm expecting to get smaller and smaller percentages of carbon. Here I have 0.8% carbon, which means almost nothing. I can't measure that, so I might switch to a different metric. If I switch to uranium, I also have 0% because uranium just has this huge half-life and it's just not it's not practical in this situation. So we're going to use a different isotope. It starts out with the option of 100,000 years, which seems good enough because I have about 75% left in this human skull here. So I'm going to drag this till it says 75. Oop, I was closer there. 75.3 is close enough, and it says 41,000 years. And it liked that answer. As you move further and further down, these metrics need to get larger and larger. So instead of 100,000 years, this one seems to be responding to 100 million years. It may also respond to uranium. It has 97.9% uranium left. So as you move further, those numbers will get larger and larger because we're getting into older and older fossils. All right, now there is a thing called a molecular clock. This is different than the clock that I was talking about at the beginning of the lecture. A molecular clock is a way for us to track or observe populations through very long periods of time. And so if there is a common trait that's maybe neutral, it's neither beneficial nor detrimental, these traits tend to stick around. They just tend to keep being copied over and over and over again with no consequence to the population. For instance, humans have either attached or detached earlobes, and this makes no difference for survival or reproduction. And so it just kind of sticks around. Some people have attached earlobes and some people have detached earlobes. There's no selection pressures to push us away from that. And so you can easily track this trait through generations and generations, and you would reliably expect those traits to stay the same. In this way, we can develop a molecular clock where we can look at the differences in the DNA or the amino acids of an organism over long periods of time. The amino acids make up their proteins. Usually we use neutral characteristics to measure this. And what's funny about these is sometimes these neutral things change very regularly. They mutate very regularly. And so maybe this year we have 80% of our population with attached earlobes. And if I come back in 100 years, we might have 80% detached earlobes. If I come back in another 100 years, I might get attached earlobes again. I might go back and forth. These things are specific to the organism that the scientist is studying, but I think it's really interesting that we get to use these neutral characteristics or these neutral adaptations to measure a population over hundreds of thousands of years. Now, if you are logged in to the Pear Deck, you might run into a geological time scale here where you just scroll and scroll and scroll and scroll, but I don't think it's linked. What I wanted by my students to scroll and scroll and scroll is just to really understand how large this history is. It's an immense amount of time, over 500 million years. And it's divided into eons, eras, periods, and epochs. And we are right up here at the very end of this Holocene. So we're way, way, way up here. Whereas sharks, for instance, they kind of showed up around this area. We've got trees popping up in the Devonian, which leads us into the Carboniferous period. And then, of course, we have all dinosaurs here, which is a super catch-all general term because you see the Mesozoic era is just huge. It's over 100 million years. And we tend to think that like T-Rexes, which might have been in the Cretaceous, and Brontosaurus, which might have been in the Jurassic, we kind of attribute all dinosaurs to this Jurassic period thanks to Jurassic Park. But we actually have three distinct periods, the Triassic, Jurassic, and Cretaceous, which had unique dinosaurs in each of those time periods. And then we get into where like everything was giant. So everything was giant kind of around here in the Ice Age. So geological time just made changes over epochs, over millions of years. Another thing that we use to measure this history of life is evolutionary stasis. This means we do have some organisms which we consider living fossils. These are organisms which look and appear to be exactly like their ancestors. So we used to think that the coelacanth was extinct. We had coelacanth fossils that were hundreds of millions of years old. So we were like, yeah, these coelacanths must be extinct because here's this fossil that's, 100 million years old. There's no way that this animal is still living. And lo and behold, in 1938, some fishermen found this weird looking fish. Good for them, they had enough foresight to bring it to a local university, and the coelacanth was identified. Now we know that there's several different species of coelacanth, but they all have this kind of prototype body where they have this broad tail fin. They're very interesting animals if you'd like to look into them, but this is just an example of an organism that didn't need to evolve anymore. It has already met its niche. It's able to survive and reproduce without really any problems. And so this coelacanth has led to other discoveries as well. One of the types of living fossils that I really like is sharks. Now, I'm apologize because this is kind of cut off in the presentation view, but if you are looking at this slide in the slideshow view, you should be able to see it very easily. Sharks are kind of perfect. They are apex predators and they have not changed in 450 million years. They pretty much started out with the perfect body type, which is this nice sleek design, lots of sharp teeth. They're fast. They can smell their prey from far away. They're evolutionary winners. And we actually have modern sharks today that resemble these ancient sharks. We even have modern sharks today that we hypothesize, such as the Greenland shark, can live hundreds of years in one lifetime. So the Greenland shark is estimated to be several hundred years old, and it's living, it's alive. Now, if you would like to, you can watch this video by Lindsay. I like her. I think she's fun. She knows a lot about animals, but you can view this in your slideshow. All right, so here we have that picture again where we can see all of the different time periods, like the Cambrian when, life just kind of exploded, and the Devonian when we see trees pop up, lots and lots of trees. We can see the Triassic, Jurassic, and Cretaceous all organized right here. And then we can see like these giant organisms here, and humans showing up way at the top in the Holocene. Okay, so we're way, way, way up here. We do see several different major extinction events. After each extinction event, we see an explosion of biodiversity. And it makes sense because think about it. Life is competing for food and resources and shelter and mates. Imagine if half of your competition dies. If half of your competition dies, then that opens up all new possibilities for life. And say we have two very similar lizard-like creatures, and one of them eats flies and one of them eats fungi. I don't know. Imagine if the organism that eats fungi dies. Now the ones that are left over have two food sources that they can eat. And this leads to an explosion of biodiversity. We can see that more in this image, even though this one's not as attractive as this one. We see this measuring biodiversity. This doesn't measure populations, it measures biodiversity. And so after each one of these extinction events, we see an explosion of biodiversity. Of course, here we have sharks showing up, and then later we have trees. Here we have the Carboniferous era, where all the oxygen made from the trees starts to kind of change how life works. And then we have the dinosaurs in the Triassic, Jurassic, and Cretaceous periods. Now, this is the extinction event that killed the dinosaurs. We believe it was a large meteor, and there's many reasons why we believe that. One reason is that there's a huge crater in the Yucatan Peninsula. Another reason is that in these layers, we have other clues about things that are going on. And all over the entire world, there's this layer of ash right around that late Cretaceous period. And so we hypothesize that that layer of ash, that thin layer of ash that we find over all the whole Earth, must have been airborne ash from that meteor shower that killed those larger organisms. But let's look back at this image. Notice the difference between this extinction event and this extinction event and the biodiversity that was still available. So even though we imagined this to be one of the worst extinction events in history, we did lose lots and lots of biodiversity. A lot of biodiversity still remained. One of the things that remained was small mammals, small rodent-like mammals, which then developed into all mammals on Earth very rapidly. As you see, we have the largest explosion of biodiversity in the history of the Earth. So today we have more biodiversity available than ever before. There is also an interactive version of this chart. We have all of these extinction events, and you can click or hover and find out more about each of these time periods, such as the Cretaceous and the Triassic, if you want to know more about these periods. I find the Devonian and the Carboniferous to be really interesting because the Carboniferous is rich deposits from those trees. Those trees had to grow and die in order for this to happen. So really interesting how the Earth's history is connected to each other. All right, next we have to switch gears and we have to talk about how do we organize all these organisms. So we found all these fossils and we have all these animals that we've identified, but how do we organize them? Well, we organize them with phylogeny, which is just a fancy word for this phylogenetic tree. The idea of phylogeny is that we put organisms on branches based on relatedness. So for instance, we have trees grouped over here. We have angiosperms are different than gymnosperms, but they're still pretty related and they probably had some common ancestor at some point. Here we have, let's see, ciliates and amoebas. Ciliates and amoebas aren't super closely related, but at some point we probably had a common ancestor. And so that's what these phylogenetic trees suggest, is relatedness. We use phylogenetic trees as a way of organizing and classifying data. Now, some biologists like to use DNA or even RNA to classify these organisms into these different categories. So here we have bacteria, archaea, and then eukaryota, which encompasses any eukaryotes with a true nucleus, such as animals and plants. They're over here in eukaryota. But phylogenetic trees used to just kind of be made up. People put things wherever they felt like based on how they looked. So now, as more genetic research is coming out, we're able to reorganize these trees into a more accurate image of how related things are. If we click on this image, then we get to an interactive phylogenetic tree, and you can kind of play with it and see how related these organisms are and where the next most common ancestor is. So if I'm looking for a common ancestor between a dolphin and a chicken, it's right here. The most recent common ancestor would be right at this intersecting point. If I wanted to know how related yeast was to a plant, potato, then that would be the most common ancestor right there. And this is all based on genetics. But that's just fun to play with. Let's go ahead and move forward and look at some different information. So phylogeny is the evolutionary history of a species or a group of species organized based on their most common ancestors. In a phylogenetic tree, the branches represent a lineage. The branch points like this represent a common ancestor. So this would be the last common ancestor between this organism and this organism. I know this image is fuzzy, just bear with me. The root of the tree represents the last common ancestor or the first common ancestor between all of these organisms. And then the tips of the branches represent extant species. Extant is the opposite of extinct, meaning these are living organisms. So let's look at the wolf and the coyote. Here I have the wolf, Canis lupus, and the coyote, Canis latrans. They have probably a common ancestor at some point. Now, there is some things that the phylogenetic tree does not assume. It does not show that one of these organisms evolved into the other. They are cousins, they are not descendants. They are descendants of the same common ancestor. So one branch went this direction, the other branch went this way. The other thing it does not suggest is it does not suggest time. So all of these branches are pretty much the same length. There's no different length of these branches. It's just organized by, we assume there's a common ancestor here somewhere. There's no assumption about time. We can't make an assumption about how old this ancestor is versus this one, okay? So time is not considered when we're talking about phylogeny. Now, the other thing that we can't assume is we really can't assume relatedness based on visible features. I already mentioned that a little bit, that scientists used to organize this information based on visible features. But for instance, marsupials found in Australia and placentals found in North and South America may have very similar traits. Look at this anteater. Okay, so this organism is a medium-sized mammal that eats ants and has a long snout and a long tail. And this animal is a medium-sized mammal with a long snout and a long tail that eats ants. So if I were drawing a phylogenetic tree, I might put these guys right next to each other and assume they have a common ancestor that's pretty recent. But actually, this is a marsupial and this is a placental, which means their common ancestor is much further back than I might initially assume. These are due to analogous traits, okay? That means that this anteater developed its ability to eat ants and its long snout and its sticky tongue completely separately from the marsupial. They both ended up at the same place, but from a completely separate lineage. So be careful. We're not organizing information by the way they look anymore. Now we're relying more on genetics. All right, so what does a phylogenetic tree mean? Well, the root is going to be the common ancestor, and the tips are going to be the extant organisms, and any branch points are going to be a common ancestor between those two tips. It doesn't tell us time. It doesn't tell us anything like that. It just tells us where our most common ancestor is. Now, there are two different main types of phylogenetic tree. Something that's called rooted means that there is an assumption about a common ancestor. So this would be the root right here. It's a little bit upside down compared to this one, but this would be considered the root. This is an unrooted version. It doesn't really make an assumption about who the origin of species is. So the phylogenetic tree could have started out with C and then developed into A and then AB, okay? Could have done that. Or it could have started out over here and branched out this direction. Or there could be some mystery organism somewhere in the middle that we haven't discovered yet. So more and more I'm seeing more unrooted trees. For instance, this is an unrooted tree. We're kind of making an assumption about the organism origin here, but we don't know for sure. Sorry, it keeps clicking on to these extra interactives, which are fun, but I want to stay on task. Okay, so rooted means we are making an assumption about a common ancestor. Unrooted means we are not making that assumption. Now, there's another type of phylogenetic tree called a clade or cladistics. This means that we are looking at a shared trait that all of these organisms share. So as I move down the line, I'm going to identify different features. So for instance, this one is looking at jaws, which means anything to this side of that identifier should have a jaw. Sharks, they have jaws. Lizards, they have jaws. Chimpanzees, they have jaws. So everything on this side of that identifier has a jaw. Everything to the right of this identifier must have hair, which means we've got our mammals over here. But not all mammals are the same. When we are talking about these two organisms, neither of them have a tail, but only one is bipedal, which is humans. That means we stand on our feet. Now, when we're using a cladogram or using cladistics, they still follow the same rules. The tips are going to be extant creatures or the creatures that we're observing. The lines represent the evolutionary history that is unique to that organism. And then the branch points represent a common ancestor. So this does make an assumption that there is a common ancestor between all organisms that have lungs, or a common ancestor between all organisms that have hair. Now, if I were to look at this cladogram as a whole, what is the characteristic that we're studying for all of these organisms? Well, all of these organisms are vertebrates, meaning that they have a backbone. So these are all chordates. What we're looking at here is a cladogram of all chordates, or anyone with a vertebrae. So this is asking us to draw a phylogenetic tree. If I were to draw a phylogenetic tree, I would think about relatedness. So here I see primates and rodents. I'm paying special attention to them because I know that they're mammals. So they probably have a common ancestor right about there. Crocodiles and birds, just so you know, are pretty closely related. Crocodiles are more closely related to birds than they are to, say, amphibians. So they're kind of in their own class. Amphibians and reptiles would be kind of studied together sometimes. It depends, but there should be a common ancestor between crocodiles and birds right there. We should also have a common ancestor between crocodiles, birds, and amphibians. So we got our common ancestor here. Now, I'm not quite sure where these rodents should fit in, but it might become more clear as we go down the line. We know that sharks and fish are related. Sharks do not have a bony skeleton. They have a cartilaginous skeleton. That means they're squishy. Whereas ray-finned fish have bones. So they're pretty closely related. Okay, and they should also have a shared common ancestor with amphibians, crocodiles, and dinosaurs. So the only thing that I'm wondering about is where to put the mammals. And so we're just going to kind of click it in there, and this will become more obvious in the next couple slides. So the difference here was the amniotic egg. So that makes more sense on this slide. All right. So what did we learn by trying to make our own phylogenetic tree? Well, we learned that it didn't necessarily matter whether we put this line here or if we put it over here, because we still end up with the same result. So we can switch these two groups if we want to. There are two descendants split from the same rod are called sister groups. So this is a sister group. And it wouldn't matter if I drew this as CBA or if I drew it as CAB, because the sister groups are somewhat interchangeable as long as I don't change that most common ancestor. Okay, I can switch sister groups very easily. Here is an example. So if I have a moss, a fern, a pine, and a rose, I can switch these two sister groups and end up with moss, fern, rose, and pine. And I have not changed any of the information. So the sister groups are pretty interchangeable. I can even change the larger sister group and switch where the fern is. So here the fern is on the end, and I still have not changed the common ancestors between these two. So it doesn't matter whether I switch those or not. Now, this is asking us to identify the unique history of the amphibian with a red line. So I'm going to find my amphibians, and this is all of their unique history. Now I'm going to stop right here because this is no longer unique history. This is shared history between all of these organisms. Then it wants to identify a common ancestor between rodents and fish. So I got to follow the rodents down the line and follow the fish down the line. Where does that intersect? Right here. This is where the blue X would go. I don't have blue, or I probably do have blue, but I'm not going to go get blue. All right. Now, when I'm talking about a clade is a group of organisms that have a similar trait. So when I'm talking about a clade, I have to gather the tips or the ends of the tree. So I can call all of this a clade, or I can call all of this a clade. But I can't call this a clade and exclude this area. So that is not a clade. Also, I can't grab the ends of sister groups. That's not a clade. I have to either choose all of one branch, or I can choose both branches, but I can't just choose half of one and half of the other. Clades can also be nested, meaning I can have a clade within a clade. What this kind of means is all primates are mammals, but not all mammals are primates. Okay? All primates are mammals, and all mammals are vertebrates or chordates. That also means that all primates are chordates, but not all chordates are mammals or primates. So we have some chordates over here like fish that are not mammals. So we can have nested clades. For instance, what is the relationship between mammals and vertebrates here? And the answer would be that all of these mammals are a vertebrate, but not all of these vertebrates are mammals. We also have something called an in-group. I know it's a lot of vocabulary, but bear with me. The in-group is just the group that we're interested in studying. The out-group is everybody else. So it says circle the clade that relates to hair. So these guys would be the in-group. They would be the out group. Okay, we're not studying them. We're only interested in the clade with hair. So this is our in group. Now, there are different types of phylogenetic trees. We've already gone over a phylogram and we've already gone over a cladogram, but what is a dendrogram? Well, a dendrogram does something unique that phylograms and cladograms do not do. Dendrograms can assume geological time. Where all of these branches are pretty evenly spaced, notice that these branches are super long while this one's super short. That automatically gives me a clue that we might be looking at a dendrogram. Here, again, I see a really short branch and then a really long branch. If I were just trying to space everything out evenly like a phylogram, it would look like this. So a dendrogram is going to make an assumption about time or evolutionary time. So here in this image, it's asking you to draw a line connecting the correct answers. The first one says cladogram. If I'm looking at a cladogram, boom, that's my cladogram. I'm looking at shared characteristics. They usually have this triangular shape to them. Phylogram means I cannot make an assumption about time. I'm going to choose like one of these that's really evenly spaced out, like this one, okay? The last is a dendrogram. That means that there is time. And look what I have here. I have a time scale. I also can tell that it's a dendrogram because sometimes I have really short branches and sometimes I have really long branches. So the dendrogram is right there. Now, why do we need to know all of this? Well, it's really just so that we can organize the information that's in our head. Like, I need to know what I'm going to call all of these turtles. And I need to kind of understand, are turtles the same as lizards? Are they the same as birds? Or how similar are they? See, crocodiles are kind of related to reptiles, but they're not necessarily the same. Crocodiles are more closely related to birds than they are to snakes and lizards. So they're kind of in their own little group over here. Um, and so we have this system, the Linnaean system, uh, described by Carolus Linnaeus, and he decided that everything should have two names called binomial nomenclature. Bi meaning two, nomial meaning number. So two numbers, nomenclature means name. For instance, the common name here is the pebble crab, but if I were looking at the scientific name, it would be Xanthias lamarcki. Now, there are some specific rules with Carolus Linnaeus' system. The first name of the binomial nomenclature should always be the genus, okay? That's going to be the description of the genus, okay? This is the second most specific descriptor of an organism. The second name should always be the species. I remember species is most specific. Species is most specific. So this is telling us the most specific information. The genus should be capital and the species should be lowercase. It should all be in italics. So it all should be italicized. That's an I. I don't know why it looks like that. I'm sorry. All right, so let's look at some different examples. For example, a tiger is a member of the genus Panthera, but Panthera also includes other big cats like lions. So I need something a little bit more specific so that I know exactly what I'm talking about. So when I talk about tigers, I'm going to say Panthera tigris. Just like when I talk about humans, I can say Homo sapien, and that should be italicized. All right, Linnaeus also made this system of classification called taxonomy, where he starts out with the most general term and then it funnels. It's just like a funnel, it funnels into the most specific. So the broadest category is going to be kingdom. Kingdom. In this case, we're looking at kingdom Animalia, which is all animals. It's such a broad term, though. Not only does it include things like crawfish, but also jellyfish, sea anemones, spiders. It's just way too general. So it doesn't give me very much information. I got to go to the next most specific step, which is the phylum. In this case, the phylum is Chordata. As we know, chordate means cord or the spinal cord. All of these organisms have a vertebra. So this is all vertebrates. Still super duper general. So I'm going to go to the class. In this case, the class is Mammalia. That means anything with hair. If it has hair, it's a mammal. And so I've narrowed it down a little bit more, but still not enough. So I'm going to go to the order, in this case, Carnivora. That includes things like bears, cats, and dogs. And I don't really want to be that general with it. So I'm going to go to the family. The family gives us a good amount of information, though. Like we now know that we're talking about canids. So here we have all of the canids, the dogs, wolves, coyotes, foxes, all of those. But I want to know specifically which canid I'm talking about. I'm talking about the genus Canis, which kind of eliminates the foxes there. And if I want to be the most specific, I have to include the genus and the species. The most specific is the species. And I end up with Canis lupus, which is the specific identifier for wolves. Now, above the kingdom, there is a broader classification called the domain. And the domain is just three big groups, the archaea, bacteria, and then eukaryotes. You notice we have kingdom plantae, kingdom fungi, kingdom animalia, and then pretty much everybody else is known as kingdom protista. And then all of these are going to be the moneras. They're just really tiny. There's lots of biodiversity there, but we tend to stick with studying these bigger animals and plants because they're easier to study, to be honest. But this is still really interesting. We are going to be doing a lot of studies over the domain Eukaryota. Okay, we're not going to study bacteria as much for our own personal reasons. They're really small and hard to see under the microscope, but also just because animals and plants are more fun. All right, based on the information that we have learned in this unit, not just this week, but last week, do we think that humans are descendants of chimpanzees? Now, humans and chimpanzees have about a 98.8% similar DNA or similar genome. But does that mean that we're descendants from one another? No, it doesn't. It just means that we both need genes that develop proteins for muscles. We both need genes that develop enzymes for our stomach acid. We both need genes that do all of these items. So we do have very similar DNA. We also have pretty similar DNA with a lot of other organisms, not quite that high though. So chimpanzees are most closely related to us. But if you look at the evolutionary history or what the fossil record suggests, is that we have some really close common ancestors, like the Neanderthals. Now, some theories say that we might have killed all the Neanderthals because we're, kind of territorial, but other data suggest that we might have interbred with them. So who knows? We also have other humanoid groups like these over here, which also we may have killed. They may have died out naturally. We don't know because they're not around yet. But these nodes are what suggest a common ancestor. So we have a common ancestor here. And if we look all the way down here, this is our common hominin ancestor. So chimpanzees may be a very, 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 very far removed cousin, not our evolutionary ancestor. All right, what if we find an organism and we don't know what it's called? Well, if we find an organism and don't know what it's called, we can play a game, kind of like Guess Who. So if you click on this, you will get to a Guess Who. And when I'm playing the Guess Who game, I'm going to ask very general questions to try to eliminate people as quickly as possible. All right, so I'm trying to guess what my character is. I'm not going to immediately ask a question like, hey, is your character going to be Jenny with glasses and pink hair? No, I'm going to ask really general questions and try to eliminate about half of these characters all at once. So my first question might be, hey, is your character a male or a female? Or hey, does your character have glasses? Because I'm much more likely to eliminate as many options as possible by asking those broader questions and then becoming more specific. So, is my character a male? I've already eliminated a lot of these answers. No, okay. So I've already eliminated all those options just by asking a general question. And now I've narrowed it down to just these ladies that are left. And I can pick a different characteristic that many of them share in common, but many of them don't, such as glasses, like I mentioned earlier. So keep this game in mind as we move forward. So this in science is called a dichotomous key. Okay. In layman's term, you might just say guess who, but in science, we call it a dichotomous key, meaning we have two parts, either a yes or a no. And that's it. Those are the only options. So when you're using a dichotomous key, they're usually very, very specific. So I can go out and buy a dichotomous key for aquatic insects living in Southeast Texas. And the person who wrote these books are usually science nerds like myself, who work at the university or so, and they catalog insects that they found. Okay, so they've done all of the work for you. And when you come to them with a specimen, they're just going to crack open this book and ask you yes or no questions. So, for instance, in this plant, does it have a narrow leaf? Yes or no? If it has a narrow leaf, does it have a bell-like flower? Yes or no? If it does have a bell-like flower, we're talking about a bluebell. So very quickly we can go down the line and eliminate all these other answer choices. So let's do an example. We're going to choose one of these little creatures. We chose this one and we want to know what is his name. We have to use the dichotomous key to find out. The first question says, does the creature have two eyes? If it's yes, we're going to go to step 2. If it's no, we're going to go to step 5. And our answer is no. We only have one eye. So I'm going to go to step 5. All right. I don't have to worry about steps 2 through 4. I'm going directly to step 5. Now, the next question is, does the creature have one or more antennae? Well, I have 1, 2. So the answer is yes here. I'm going to go directly to step 6. I don't even care about this question because I'm going to step 6. All right, in step 6, it says, does the creature have one antenna? No. Okay. Does the creature have 2 antennas? Yes, it does. So I know that his name is Carl. Nice to meet you, Carl. We can do this same activity with leaves or insects or really any organism. That's why it's important to learn about the dichotomous key now, because in future labs, we will be using the dichotomous key regularly. So if I'm trying to identify this second plant down here, I'm going to go through my dichotomous key. The first question is talking about needles. Neither of these leaves look like needles, so I'm going to go with non-needles, which means I'm going to step 3. I'm going to skip steps 2 because I don't care about that. Now in step 3, it says, is it a simple leaf or a compound leaf? It is a single leaf that's not a compound leaf. If you want to know what a compound leaf is, this is a compound leaf. Okay, we're not using a compound leaf yet. So it's a simple leaf. I don't care about step B. I care about A. It says go to 4. So I'm going to go straight to 4. Does it have a smooth edge or a jagged edge? Well, this is a smooth edge. So I'm going to go to step 5. Step 5 says, is the edge smooth or lobed? Well, that may be a little bit ambiguous because we just talked about smooth. So if I were writing this, I might find a different word to use there, but this is a lobed leaf. It has these large lobes here. So I can safely assume that this is a white oak. If I were to do the same thing for the top one, I would get magnolia. This is a magnolia leaf. If you would like to practice, you can do these as well. Just go through the dichotomous key and answer the questions yes or no, and then follow the directions. Okay. Thank you for joining me this week. I know it's been a little bit of a long lecture, but hopefully very informative. Don't forget, you have an opportunity for extra credit by completing the optional chapter homework. You watch a video and then do a drag and drop activity. If you would like to check your answers, the SI leader will have that answer key. They're not supposed to give it to you though. You're supposed to find those answers on your own, and then they can help you check your answers. So don't go to the SI leader just expecting them to do your work for you. They're there as an assistant. Also, don't forget to do your chapter laboratory write-up. Print this out, scan the QR code, and then complete the activities. There will be a 5-question open note lab quiz in your lab Blackboard. But that doesn't mean that you should neglect the lecture quiz. There is a 10-question lecture quiz every week in Blackboard. So make sure that you take care of those. If you need further information about this week, then you can go to further reading, which is the textbook, or do some vocabulary exercises by clicking on the Quizlet. We also have vocabulary-based games and a Quizlet, which is located in the unit lesson. I hope you all have a great week, and I will see you next week. Please don't forget that next week is going to be your exam. We don't have any new content being assigned next week, but you cannot neglect to take that exam. It is due by Friday at 5 p.m. of next week. So make sure that you take that on time. This is not an assignment that you can turn in late. It has to be done by the specified time, which is Friday at 5 p.m. All right, y'all have a great week. And if you have any questions, feel free to reach out to me. I'm available by email or appointment. Thank you.